I actually took a few slightly different things away from ASCO and EHA, and I've just tried to summarize them into two buckets. So one is about high-risk myeloma, and then the other section I'm going to talk about is real-world data. And I know in the last session with Martin and Katia, you spent quite a lot of time talking about high-risk myeloma. So hopefully a little bit of repetition is good, and hopefully I'll maybe bring out a few other interesting things. And it's really to say at these big meetings, as well as learning new data, we also spend a lot of time looking at all of the data, putting it all together and discussing and educating each other. And at ASCO, one of the kind of general sessions which people were encouraged to go to was really a review of high-risk myeloma, and particularly what is high-risk myeloma, why it's important to identify patients with it, how we currently treat patients with it, and how we can improve on that. And I think it came up in the talks earlier that this is not just a problem that patients struggle with, it's a problem that doctors struggle with as well, particularly because the definitions seem to be changing quite often, and that's really because we're getting much more data about patients and we're understanding it much better. And so there was a session talking about this, and I just wanted to share some of the kind of take-home messages from that. And so the first one was really discussing how we diagnose it now. And I think, as Jenny said, we do that we use this RISS, which is a combination of 2 blood tests, and then a combination of looking for some very specific genetic abnormalities. And those are abnormalities within the myeloma cell, not within the kind of whole patient. But what's become evident is that this is a good, it's a good way of doing it, but it's maybe not specific enough. It doesn't really home in on all of the patients we want to think about as being high risk and maybe not responding to treatments as well as we want them to. And that's because some patients can be identified as high risk with this test and actually don't have high risk myeloma and do very well with things, whereas other patients can be not identified with this test and actually don't do as well with things. And I think it's really important now, particularly as we've been discussing that patients are living longer, that as a doctor, when you see a patient, and as a patient, you kind of, to some extent, want to know, are you going to be one of those patients that have a good response and a long survival? Or is it actually not going to be so optimistic? And I think that's important just for a general planning side of things. And so there was lots of discussion about, okay, my slides aren't moving forward now. I really am having a traumatic time. So there was lots of discussion about what different factors related to high-risk myeloma. And as I say, people have spoken about this before, but things like maybe the kidney's not working so well, so you can't manage to have the therapy so often, or maybe being a little bit frail or not so mobile, meaning that getting to the hospital and generally tolerating the treatment so well is important. There was also about how patients respond to therapy, and I think Katia mentioned this earlier, that some patients either don't respond to therapy and take a long time to respond, or indeed their response to therapy is actually quite short. There was then lots of discussion about the things we often talk about. So when we can find plasma cells in the blood, or when we can find lots of myeloma in the bones on the PET scan, or indeed if there's lumps and bumps of myeloma. And then I think, again, people have been talking about this. There was lots of discussion about some of the genetic abnormalities. And I find this in a lot in my own practice. I guess it's because this is a subject quite close to my heart. But often patients are told that they have a high-risk abnormality, but it doesn't necessarily mean that it is a high-risk abnormality because, as I say, the technologies have changed and it's a very rapidly moving field. And so there was just some discussion about the nuances about how we define these different abnormalities. And the reason that was really important discussion to have is Because up till now, we've always put all patients together in the clinical trials, and we've heard a lot about this today. And in this diagram, I'm trying to point out the kind of what we call the standard risk patients who are in purple, and the high risk patients who are in blue. And we put them all together in a clinical trial, and we treat them with different treatments, and then we analyze what happens. And then at the end, we kind of say, oh, by the way, what happened to the high risk patients? How did they do? And that's an okay way of doing things, but there's probably not actually enough patients in that group to really make firm conclusions. And so one of the kind of general movements in the field, as well as some of the studies that were presented at both ASCO and EHA, was actually trying to pull out those high-risk patients and treat them differently, okay? And both Katia and Martin, who spoke before us, presented their data on new clinical trials and new ways of doing this. And lots of questions in the chat box have also been discussing about how maybe these patients who we know maybe don't always have a very long response to daratumumab or RVD or whatever we're going to use, maybe we should think about trying to treat those in a slightly different way with a CAR T cell or with a BiTE, and those studies are just beginning. I'm not suggesting if you're a high-risk patient that you should just kind of go willy-nilly off down doing this. It needs to be done in the context of a clinical trial. But for me, I thought it was a really encouraging step forward that we were beginning to, I guess, try and personalize treatment for myeloma rather than just putting everybody together, try and pull out the different groups which are important. So that was one kind of important take-home message for me about how the field was rapidly moving. The other thing that I found really interesting at the meeting was this whole concept of real-world data and real-world evidence. And again, there's been a couple of discussions in the chat box about this. But at the moment in the US, when we think about all cancer studies, there's probably only about 10% of patients who make it into a clinical trial. And I'll talk about that in a minute as to why. But that leaves both the patient and the doctor saying, okay, there's this really lovely clinical trial data, but how does that apply to me as a patient? And so there was a lot of discussion at both meetings about why patients are not entering clinical trials. And that can be about the availability of clinical trials. Clearly, the US is a huge place and there may not be a clinical trial next door to you, but also about the entry criteria for clinical trials. And it also highlighted the importance of this real world data. And there was a lot of what we call real world data presented at the meeting. And I'll come to that in a moment. But the first thing was really the discussion about why not all patients make it into a clinical trial. And this was just a paper that was presented a couple of years ago that looked at clinical trials and found that most clinical trials has about 16 eligibility criteria. And that's essentially, say, kind of a checklist of what kind of patient you need to be to get into the trial. So for instance, if it's a myeloma study, some of them are obvious, you need to have myeloma, okay? But other ones are maybe more specific. So if the drug, for instance, is secreted by the kidneys, it may be that patients who don't have a good kidney can go into the clinical study. And what they discovered was that actually patients with lots of comorbidities, patients who were older and frailer were less likely to go into a clinical trial, but also potentially patients from ethnic minorities or lower social economic groups were also less likely to go into a clinical trial, either because they were unsure of what they were being offered or because those studies were not available in their area. And for myeloma patients, examples of why patients don't make it into clinical trials could be because they've got heart problems. It may be because they've had a previous cancer, and therefore that's often an exclusion criteria. And something I think that came up earlier, that their disease may be non-secretory, and that again is often a reason why patients can't make it into clinical trials. And so there was this whole discussion about how do we get data to support our use of our drugs in that kind of setting. And what we call that is this real world evidence. And I'm kind of in some ways preaching to the choir here, because Myeloma Crowd is a really good example of collecting real world evidence data, because there's a number of ways we can get the data, and one of them is from a registry. And I know Myeloma Crowd has an excellent registry, which a lot of the clinical investigators are very excited about using, and I know many patients are using it too. But there's also things like electronic health records, and then in the US, insurance claims and data. And it really gives us an opportunity to say, okay, how does this data apply to me? So I'm not saying we shouldn't have clinical trials, but yes, clinical trials are great. They're in a very controlled circumstance, and they tell us if the drug can work. Whereas this real world data is kind of in your everyday clinic, and it tells us more about does the drug work? And so some of the examples that were presented at these two meetings were examples about where, for example, CAR T-cell therapy, we've had a lot of discussion about that today, but I think as everybody knows, it's not available for everybody at the moment, and it's quite a difficult process to manufacture the CAR T-cells. And so what the companies and what the physicians have been trying to do is to use some of this real world data and say, okay, if a patient was to get a CAR T-cell, we know what would happen. But if they got one of the other treatments, what might happen? And how do we decide which is a better treatment? And it's very difficult to do that kind of analysis. But there is some data to suggest that potentially, if you were a candidate for a CAR T-cell study, then you might do better if you had the CAR T-cell study rather than if you got one of the other therapies. And there was also lots of data from the real world about vaccines, for instance, particularly with COVID. For example, we know exactly how the vaccines did in patients who don't have myeloma, but how are the vaccines doing in patients who do have myeloma? And so there was lots of nice real world data presented on that. And I don't know, those of you who are into the Twitter sphere, there was actually some updated data from the FDA this week suggesting that they were really going to take much more or look at this much closer in when they're coming to think about their decisions. So I think those were the two things I wanted to kind of put through, much more general things, but really very important and useful for both myeloma patients and doctors. So thank you.